شاء الله بتنزل القوائم الجديدة بنعتمدها بإذن الله مشان الرياض. آخر امتحان أخذته في حياتي كان medical statistics في مرحلة الدكتوراه. دخلت على الامتحان وكان الدكتور تبع المادة عراقي بأمريكا وبلاك اوت نسيت كل المعلومات كلها، يا دوب اخذ الافرج يا دوب بس. الامتحان ساعتين اني اتذكر شيء ما في فايده. اخذ لي الدكتور عنده على المكتب قاعد يحكي معي، يا مأمون يا ليش ربنا كذا مش انت بتعرف تعمل ايه؟ بتعرف طب روح منيح مره. The same thing happened yesterday with this problem. Just like comes blank. How can you solve this? Well, since you have pKa, you can, one way of doing it, you can convert it like Ka equals concentration of proton times concentration of the conjugate base, اللي هم equal to each other, so x squared, على the concentration of the acid اللي هو موجود عندهم 0.02. okay. بنطلع وقتها ايش هي الاكس اللي هي نفسها الكونسنتريشن اوف ذا بروتون، صحيح؟ اللي خلص بنعرفها وقتها. بنحسب البي اتش. هلا هون اللي صار عندنا هذا السكشن، هذا الكويشن زي اللي سالتكم اياه قبل هيك، انه صار في اديشن اوف ان اسيد، صار في سويتش، الايكوليبريم اختل، راح ليفت اور رايت. يعني what do you add? Sodium peroxide. So basically, the concentration of the protons would decrease by certain level. بعدين it goes up, equilibrium shifts. Acid would go down by this much, okay, and the conjugate base would go up by this much as well. والpKa موجود عندنا فبنحسب الpH as well. من الهندرسون هاسلباك equation. So. فحكينا على compensation او حكينا على respiratory conditions, acidosis, alkalosis. What happens exactly if someone is asthmatic, CO2 level goes up, this goes to the other side, so that goes up, but pH goes down, so this is acidosis. Okay. If someone has panic attacks, for example, this goes down, the equilibrium shifts to the right, this goes down, alkalosis, because pH goes up. بصير عندنا compensation. Compensation هلا, in order to increase the concentration of protons, what kidneys do, they decrease the concentration of the bicarbonate. So that the reaction would go to the other direction. Okay? So the reaction will go to the other direction. So this goes, eventually this would increase, and this would decrease in CO2. The thing is, CO2 is gas, and it is measured. It's measured in terms of pressure rather than concentration, molarity. That is. Okay. عشان هيك عندكم the letter P in the tables at the end. So that's why you have PeCO2, that is pressure of CO2. هلا الفول compensation زي ما حكينا لو عندنا احنا respiratory acidosis, meaning that this goes up, reaction goes to that direction, so this increases as well. Okay, this increases, reducing pH. لو عندنا alkalosis, again respiratory alkalosis. This goes down, the reaction goes that direction, concentration of protons goes down as well, pH goes up. فهي عندنا pH is up. To compensate, we decrease the bicarbonate, so now its level would be below normal, so the reaction would shift to the other side. هون صار عندنا full compensation because the pH is within normal range. Metabolic acidosis. This goes up, the reaction goes to the other direction. Again, so actually this it goes to the other direction, increasing the CO2. Now, in order to decrease, order to have compensation, CO2 must go down, so there is increase in the rate of breathing. So CO2 goes down so that the reaction would go to the other direction, and this goes down as well. So both of them would go down eventually. Same thing with the atherosis, or the opposite with the atherosis. لما يكون عندنا partial compensation, so that the pH is still not normal, it's still outside the normal range, 7.35 to 7.45. So if you have respiratory acidosis, this means that concentration of protons, respiratory acidosis, this means that this is high. It results from an increase in CO2. So this goes in here. So what we have to do is to increase bicarbonate. The kidney would intervene to increase this, so the reaction goes to that direction. So these two would go up. Okay. The main problem is that CO2 increases. The main problem is CO2 increases. The solution is to increase the bicarbonate. Yep. Respiratory alkalosis, CO2 goes down, so the reaction goes to that direction. In order to shift it back, decrease the bicarbonate. Okay? So these two would go down below normal level. Alright? Okay. هلا metabolic acidosis. Acidosis, this goes up, the reaction will go to that direction. As a result, this decreases. In order to compensate, this must actually increase. You can feel another one. This must increase so the reaction goes to the other direction. Let me fix this slide. I think there's something. Let's try to do this slide. Okay, let me fix it, and I'll send you a note على e-learning. صحيح أنا نزلت محاضرة اليوم الكربوهيدرات على e-learning وحطيت لكم نوت. امبارح حكوا معي البوك شوب. The book is now it's for sale for 24 JDs, not for 30. فعملوا discount for some reason. Let me review the slide and I'll get back to you. نعم. إذا كان متوالق compensation من اللنgs. بتقل ايش؟ بتقلل كمية CO2 عشان تروح على الدايركشن الثاني. عشان to increase the rate of breathing. Okay, let's go back. Sorry. So if this goes up, metabolic acidosis. If this goes up, pH goes down. Okay, فعرف انه صار في عندنا acidosis. As a result, the reaction shifts to that direction. ماشي؟ As a result, this goes below normal level. So it goes down. As a compensation, what the body would do to decrease the level of CO2. So increasing the rate of breathing. When this is decreased, the reaction shifts back to the other side. So CO2 goes below normal level as well. Thank you. Alkalosis. When this goes down, the reaction goes to that direction. As a result, the bicarbonate increases. لأنه الرياكشن راح على هذا الاتجاه. So the bicarbonate increases. In order to compensate, we have the lung would intervene. ايش راح يصير عندنا؟ Rate of breathing would slow down, increasing the level of CO2 so that the reaction goes back to the other side. Decreasing this so the reaction will go to the other side. عيد نعمل فورماتين ونعيد من جديد. اوكي. مرة ثانية نبدأ من هون، تعملوا delete restart. طيب full compensation. هلا عرفنا ايش لا نرجع من هون. Respiratory acidosis. The main cause for respiratory acidosis is that a person has high level of CO2 in blood. Okay? One cause is, for example, choking. Another is asthma. CO2 level goes up, so the reaction goes that direction. Hello? Okay? That results in increasing the concentration of protons. شو صار عندنا هون؟ pH is now below normal level. This is acidosis. For alkalosis, if someone is panicking, for example, this goes down, so the reaction goes to that direction, reducing the concentration of protons, increasing pH. This is alkalosis. Hello? Metabolic acidosis and alkalosis, the effect is direct on proton, level of proton, like I said yesterday. Resulting in decreasing the pH, acidosis, or increasing the pH, alkalosis. حلو؟ حلو. هلا بنرجع على compensation. I said if the defect is with the respiratory system, if the defect is with lungs, then the kidneys will fix the pH. And vice versa. If the problem is with the kidneys, then the lungs would fix the pH. Compensation can be complete or it can be partial. The difference between complete and partial is that with complete compensation, pH is within normal range. Partial, pH is not within normal range. But you look at the levels of CO2 and the bicarbonate, and they are still not normal. So you can say that the body is trying to fix the pH. The body is trying to compensate for the increase or decrease in protons or CO2. but it has fixed the problem for compensation, or it hasn't partial compensation. This is the difference between the two. Now, in case we have full compensation, notice that the pH is normal in all cases. But you can see that it's still it's still below or above 7.4. Okay? But if you look at the levels of CO2 and the bicarbonate, okay, you can see that they are not within normal range. You'll learn about the exact numbers later on in physiology. So with the respiratory acidosis, what happens again is that this is above normal level. Reaction goes to that direction. That goes up, pH goes down. So CO2 is higher than normal. To compensate, to fix the problem, we have to increase the concentration of the bicarbonate in blood. ليش? So that the reaction would shift to the other direction. So as a compensation, this must also increase as well. عشان هيك these two are up. For respiratory alkalosis, this is below normal level, so the reaction goes to that direction. To compensate, we have to decrease this, so the reaction would go to the opposite direction. So these two would go below normal range. معي إن شاء الله. This is why I said yesterday, go home, look at these tables, look at these slides, and think about them logically, so that you'll understand. Metabolic acidosis, this goes up, pH goes down. فالreaction would as a result go to that. So the reaction would go to that direction. This goes down. In order to I'm sorry. So this goes up. So that's metabolic acidosis. So the reaction goes to that direction. And this goes below normal levels as well, as a result. هلا, in order to fix the problem, okay. Sorry. ايش حكيت؟ I'm sorry.
هذا مساوي الاسيدوز.
Yes.
يعني في ارتفاع في الاسيدوز في الجسم بيسبب عمليات
في ايش؟
ارتفاع في الاسيدوز.
Yes.
بيسبب عمليات حيوية معينة.
Yes.
فالنظام رح يقلل كمية CO2 موجودة في الجسم عشان CO2 لا تأثير.
حتقلل كمية CO2. حتقلل كمية CO2. Okay. So the reaction goes to that direction, and the lung would compensate by reducing this CO2 level so that the reaction would go to that direction as well. عشان ليش؟ عشان نقلل كمية البروتونز اللي موجودة in blood. Now for metabolic alkalosis, this goes down. As a result, the reaction goes to that direction. So this goes above normal level. هلا احنا ايش بدنا نعمل؟ بدنا برضه نخلي الرياكشن يروح هيك علشان نزود من كمية البروتونز, and as a result, CO2 level goes up as a compensation so that it goes to that direction, and to increase concentration of protons and decrease pH levels. هيك صار عندي أنا compensation. هذا partial compensation because again that's full compensation now pH is within the normal level. Now partial compensation. Now تفضل. ايش السلايد اللي yes. على بس ايش؟ هو مكتوب هو التأثير على البروتون. طيب بس as a result هلا هذا when it increases this decreases as well, لأنه الرياكشن راح يروح على هذا الاتجاه, فهذا راح يقل. مرة ثانية sorry. اسمع خليها لبكرة. خليها لبكرة. I'm sorry. ندخل بالموضوع الجديد معلش. اعمل ديليت لهاي السلايد لهاي السكشن من المحاضرة. Let's get into carbohydrates for now. Carbohydrates. The definition of carbohydrates is they are polyhydroxyaldehydes or ketones. So the main functional group in carbohydrates is that they either contain aldehydes or they contain ketones. And this structure it contains multiple hydroxyl groups. So that's why they say polyhydroxyaldehydes or ketones. That's their functions. They are good sources of energy in the body. There are different structures they can be structural molecules. That is, they form the structure of, like, let's say, stems in plants. So they are structural molecules. They form a certain structure. Or chitin in insects. The hard shell of insects, for example, or certain animals is really made of carbohydrates. They're also building blocks, meaning they can be used to form larger molecules. And they're used for cellular recognition. So يعني, it means that cells can recognize other cells by looking at the sugars on the surface of these cells. And that's how they recognize these different cells. But carbohydrates can be classified according to different criteria. One of these criteria, the first type of classification, is the number of units that this sugar is made of. They can be classified into, or they can be categorized as monosaccharides, meaning that these are molecules that are made of just one single sugar entity, molecule, like glucose, fructose, mannose. Or they can be disaccharides, meaning that you have two of these monomers coming in together, forming a two-sugar molecule, like sucrose and lactose. And you have oligosaccharides as well. Oligo means short. So an oligosaccharide is basically a molecule that is made of 3 to 10 of these monosaccharides. And then you have polysaccharides, methyl, glycogen, and starch. So this is one type of classifying these sugars. Rarely that you have sugars in the body in the free form, or it's not as common as finding these sugars integrated or conjugated or attached to other molecules. So you will have sugars as polysaccharides again, or you can have them as attached to proteins. These are known as glycoproteins or proteoglycans. The difference between glycoproteins and proteoglycans is the ratio of sugar to protein. If you have a lot of protein and some sugar, then it's known as glycoprotein. If it is a large molecule made of a lot of sugar and some peptides, then it's known as proteoglycans. We'll talk about these at the end. You can have sugars attached to lipids, and these are known as glycolipids, or you can have them attached to certain groups like methyl groups, for example, and these are known as glycosides. You can have sugars attached with amino groups, or you can have the sugars even modified, molecules known as glycose aminoglycans. So a lot of sugar molecules, and the sugars, the monomers, are modified. As an example is hyaluronic acid, or you can have them as part of DNA and RNA structure. We talked about that last semester. So let's talk about monosaccharides, single unit sugar molecules. Again, you can have these sugars as aldoses or ketoses. So to name a sugar, it has to have this O's at the end of the name. So once you have this OSE, glucose, sucrose, fructose, you know that we're talking about a sugar. And these sugars can either be aldoses or ketoses, meaning that they either have a functional group as an aldehyde, or they can have them as a ketone. And usually you can find them in books drawn something like this. This is Fisher projection, and it means that these are extended forward to the front of the molecule, and these in green are they are oriented to the back of the molecule. طب ايش هنعمل؟ عندنا كمان النمبرينج. Numbering of carbons is important. Since this is the main functional group, this would be carbon number 1. And usually, actually always, it's drawn when it is in a chain structure. It is known where you have the functional group to the top of the molecule. بعدين you have number 2, you can have more than that, so you can have n number of carbons, and at the end you have CH2OH in both ketoses and aldoses. Now if you have a ketose, the ketone group must be carbon number 2. And again, it has to be on top of the molecule. Now whenever you have these bonds right here, all these bonds are again extended backward, and these are extended inward, forward. ماشي? So that's basically the simple structure of monosaccharides. These are the common monosaccharides, and you have to know these structures. Yeah? You have to memorize the structure of some of these sugars, not all of them, but some of them, and I'll let you know what you need to memorize exactly. Glucose is the most famous sugar, right? It has mild sweet flavor. It's not really that sweet. It's the blood sugar, main source of energy, found in all disaccharides and polysaccharides. This is the structure of glucose. You can have galactose as well. This is galactose. It doesn't taste that sweet. Fructose does, and that's why you find fructose in a lot of products. يعني إذا بتروح, if you buy gum, for example, or syrup, look at the ingredients. It would be written high fructose syrup, because fructose is very sweet, and so it's used in different products. This is the structure of fructose. Now I want you to notice the difference in structure between glucose and galactose. We'll talk about that later on, so pay attention to that. So these are the aldoses, and these are the structures that I want you to know. So the simplest aldose is a sugar known as glyceraldehyde. So this is an aldose, a 3-carbon sugar. Then you have ribose. You should know what ribose is, right? DNA and RNA. That's the structure, and it's made of 5 carbons. And then you have these 3 sugar monosaccharides that you need to know: glucose, galactose, and mannose. So you need to memorize these structures. In terms of ketoses, the most famous is fructose, which is right here. Okay, this is the simplest ketose, and it's known as dihydroxyacetone, a 3-carbon sugar. Okay, so that's how we can classify sugars. Another classification is by the number of carbons that this sugar contains. Notice that the simplest sugars, there are 2. Glyceraldehyde, dihydroxyacetone, they are composed of 3 carbons. And that's why they are known as trioses. Again, O's for sugar, tri, 3. So trioses. Now, if you have 4 carbon sugars, then they're known as tetroses, then pentoses, hexoses, heptoses, and so on. So according to the number of carbons.
دكتور الستركشرز اللي لازم نعرفهم اللي محطوطين في المصطلح.
الايه؟
الستركشرز اللي لازم نعرفهم نحفظهم
محطوطين. They are square. The structures you need to know, they are placed within a square. Okay? So, like fructose, for example. These are the ones that you need to know. All right. So let's go into trioses. We have 2 simple sugars, dihydroxyacetone and glyceraldehyde. This is the structure of these 2 molecules. Now, notice something. If you look at glyceraldehyde, it has a chiral carbon. Question is, what is the definition of a chiral carbon? It is a carbon that is connected to 4 different groups. That's what a chiral carbon is. Dihydroxyacetone does not contain a chiral carbon because it has this carbon that forms double bonds with oxygen. So it does not contain a chiral carbon. Hala, what's the importance of this chiral carbon? Well, hala, we'll talk about it. So look at these structures right here. Is this molecule a chiral molecule? Is this carbon a chiral carbon? Yes or no?
No. Yes.
هذا الكربون
Yes.
chiral. Okay, مين حكى له؟ قلت له. Cool.
مع الثاني.
How about this one?
No.
Ah, Captain. No. Okay. This one here. Yes. This one here. No. طب نطلع على هذا المولكيول. Let's look at these carbons. Is that a chiral carbon? Ah, very fast. Chiral. الآن group غير عن هذا الجروب غير عن هذا الجروب وعنده هيدروجين. So it's chiral. طب هذا يلا يلا معي رول دول. هذا لحظة طلع هذا
No.
هذا chiral.
No. Acre. Acre.
مين بيحكي اياه؟
No.
طيب. فهي الانسرز هون chiral A chiral chiral A chiral هذا chiral هذا chiral بس not chiral لأنه it's attached to two methyl groups. Okay, كثير كويس. نيجي على هذا flow chart. Let's look at this. So we can place these carbohydrates in different families according to their structures. We can say that they can be isomers. What does isomer mean? It means that these, if two molecules are isomers, it means that they have the same number of carbons, hydrogens, oxygens, nitrogens, whatever, but they're connected differently to each other. So that's what isomers mean. Now these isomers, there are two different types. They can be constitutional isomers or they can be stereoisomers. Constitutional isomers or constitutional isomers means that they are connected differently, the groups, but they have the same number of groups. Now, stereoisomers means that they are connected the same exact way, except that they are oriented in space differently. لأنه إحنا we think in 3 dimensions. So, for example, you have two molecules, and you have a carbon that is attached to a hydroxyl group. And for one molecule, you have this hydroxyl group oriented, for example, forward. For the other group, it's oriented backward. But they're connected to the same exact carbon. Does that make a difference? Huge difference. In terms of biochemistry and our body, huge difference. It can make something that tastes sweet, if you change its orientation, it can taste really bitter for certain types of food. So the orientation of these groups is important. So that's the difference between the two. Now, one of these 16 molecules is glucose. Your homework is find out, and after going through the first few slides, after learning what glucose looks like, you need to pick it out of here. Now these are all isomers. They have the same number of carbons, hydrogens, and oxygens, but they are connected differently in space, and that's why they are isomers. So you can see that, for example, you have all of these hydroxyl groups oriented to the right. You have this one here with a difference in the orientation at this carbon. The hydroxyl group is oriented to the left. Again, these are all different molecules. So you need to find out, search for glucose, galactose, and mannose as an exercise. So how many isomers can a molecule make? It depends on how many chiral carbons it has. And the basic formula is it's 2 to the n. N is number of chiral carbons. So we have 16 here. So how many chiral carbons does this molecule have? 4. So, okay. So actually it is 4. And we will look at it شوي in more details. So these are the different isomers, stereoisomers. Now you can have some of these stereoisomers, they can be enantiomers. What does enantiomer mean? It means that these two isomers, stereoisomers, are mirror images of each other. That's basically what it means. So as an exercise, you can go back to that slide and you can, for example, look at this structure and pick its enantiomer. Now each molecule should have just one enantiomer. You do not stand in front of a mirror and you see two people. If you do, immediately run to a doctor أو psychiatrist. Okay? معناها هلا أنتم رح تشوفوا 10 قدامكم. So each molecule should have one enantiomer, one mirror image of that molecule. Okay. So that's what the enantiomer, that's what the enantiomer means. Now, for glyceraldehyde, it has its mirror image. And to differentiate between glyceraldehyde and its mirror image, they're given this designation right here, L or D, depending on how they absorb light or reflect light. That's organic chemistry. I'm not going to get into that. But L means level and D dextrose. To me, as someone who is somewhat illiterate in organic chemistry, I say L means left. ليش? And it makes life simple for me. ليش? لأنه what we're doing is we're looking at this chiral carbon and we're looking at the orientation or the direction of this hydroxyl group. And so if it goes to the left, then I say, that's L-glyceraldehyde. The opposite is D, where you have the hydroxyl group oriented to the right. ماشي? So that's the difference between L-glyceraldehyde and D-glyceraldehyde. So go back to these 16 isomers of glucose, but we talked about actually 15 isomers. I know number 16 is glucose itself. And pick the L-glucose and the D-glucose. طيب هلا نحكي كيف الفرق بين D وال L. So we're looking at the chiral carbon. So how many chiral carbons does this molecule have? How many chiral molecules? Sorry, how many chiral carbons does this molecule have? بدي الكل يكون confident بالانسر تبعه. Two. Exactly, two. اللي هم هذا and this one. This is not a chiral carbon because it's an aldehyde, double bond O. This is not a chiral carbon because it's attached to hydrogens. So these two are chiral carbons. How many isomers can this molecule form? Four. Exactly. But for each molecule we have only one enantiomer. We have D and L. Now how do we know that this is the L and this is the D? Here's the trick. Here's the solution. You look at the chiral carbon. That is farthest apart from the functional group. Again, to differentiate between a molecule, if it is an L or a D molecule, sugar, you look at the orientation of the hydroxyl group. That is farthest from the functional group. So here's the functional group. It's the aldehyde or the ketone in case of ketosis. You look at the chiral carbon that is farthest from this functional group. So you're looking at this carbon right here. You look at the OH, it's going to the left. It's L, it's going to the right. It is D. Simple. So go back to these 16 isomers, pick glucose, and find out if it is a D or an L glucose, and pick its enantiomer as well. Got it? نعم. ايش هو؟ بس انا ما هو كيف هو يعني بيعملوها بالتجربه عشان يحددوا الاورينتيشن اوف الهيدروكسيل جروب، بيعملوا هاي التجربه. اوكي؟ بس اول ما انت بتشوفها بدك ترسمها you draw it in this way so that the OH goes to the left or right when you draw it. Okay? هلا. Now these stereoisomers I said they can be enantiomers if they are mirror images of each other. If they are stereoisomers but they are not mirror images of each other, we call them diastereomers. So for these 16 isomers that I showed you, one of them is L-glucose, the other is D-glucose. It's enantiomer. Okay? Halla. And you have all of the other ones, all 14 isomers are diastereomers, meaning that they are isomers, but they are not mirror images of glucose. Halla. So look at these 4 molecules, هذا erythroids. Are these enantiomers? Are they هذول enantiomers? Are they mirror images of each other? يلا. هذول mirror images of each other? Oh, they are. هذول الاثنين. Yes, they are. لأن هذا رايح يمين وشمال، بعدين شمال يمين. So they are mirror images of each other. Are these enantiomers? No, they're not. They are diastereomers. So they are not mirror images of each other. إذا إحنا عندنا إيش؟ عندنا الايزومرز ممكن يكونوا stereo isomers, ممكن يكونوا constitutional isomers, and we have stereo isomers. They can be enantiomers if they are mirror images of each other, or they can be diastereomers if they are not mirror images of each other. Now, to differentiate between an L or a D glucose, you look at the chiral carbon that is farthest from the functional group. So for glucose, you can pick its enantiomer. And for a D-glucose, its enantiomer would be an L sugar, not necessarily L-glucose. To pick L-glucose, you need to look only at the hydroxyl group that is linked to the chiral carbon that is farthest from the functional group. That orientation is what determines L or D glucose. Let's look at epimers. The definition of epimers is that basically they are diastereomers and they are different in the orientation of this hydroxyl group of one chiral carbon. Not everything, just one. So you look here, for example, mannose and glucose. There is glucose. Hydroxyl group, this is a chiral carbon. Each one of them has 4 chiral carbons. 1, 2, 3, 4. 1, 2, 3, 4, and so on. You look at the hydroxyl group, it's oriented to the left, to the right. So they're different. Left, left, right, 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 right. So they differ in the orientation of only one hydroxyl group. And we call them, as a result, epimers. Specifically, we say that they are epimers at carbon number 2. Look at glucose and galactose. Right, right, left, left, right, left, right, right. So they differ in the orientation of one hydroxyl group, which is the one that is linked to carbon number 4. So we say that galactose and glucose are epimers at carbon number 4. Got it? What is the relationship between galactose and glucose? Sorry, galactose and mannose. Sorry. What's the relationship? Are they isomers? Are they isomers? Yes, they are. ايش تعريف isomer? Same number of carbons, hydrogens, oxygens, etc. Right? They do. So they are isomers. Are they constitutional or stereoisomers? They are stereoisomers. All groups are connected the same exact way. Great. Now, are they enantiomers?
No.
They're not. Are they diastereomers? Yes, they are. Basic. So they're the diastereomers. Now, are they epimers?
No.
No, they're not. So we stop at diastereomers. They are just diastereomers. Yeah? هلا some naming organic chemistry. If you have an aldehyde and it reacts with an alcohol group, it forms a group known as hemiacetal. And hemiacetal is where you have a carbon that is linked to OH, OR, H, and carbon. If you have another reaction with another alcohol group, you will have an acetal group. Now, if you have a ketone and it reacts with a hydroxyl group, it forms a hemiketal, which is basically a carbon that is linked to OR, OH, C, C. So the difference between hemiketal and hemiacetal is why it is that in the hemiacetal you have a hydrogen, and in the hemiketal you have two carbons. If the hemiketal reacts with another alcohol, then it forms a ketal where you have OR, 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 C, C. So that's the difference between acetal ketal, hemiacetal, and hemiketal. Why do I say that? Why is that important? Because rarely that you will have a monosaccharide that exists in the chain structure. Rather, you will have it as a ring structure because it's more stable. So it goes into equilibrium, ring, chain, chain, ring. Except that the direction of this reaction goes from chain to ring, more than ring to chain. It's more stable as a ring structure. How can a sugar form a ring structure? Well, when you have this carbon right here, the hydroxyl group that is attached to the last chiral carbon, when it attacks the functional group. And when you have it, then you will have the formation of a ring structure known as a pyranose. كيف عندكم بالorganic chemistry you have a benzene ring, you have a phenol ring, and you have all of these different names for rings. Well, you have this ring structure known as a pyranose. Again, it's O's, so it is a sugar molecule. So that's the name of a pyranose. Now notice how it's given this three-dimensional shape by having this dark edge right in the bottom. So when you look at it, when you look at this pyranose structure, that's what it looks like. That's carbon number 1, carbon number 2, 3, 4, 5. This was the functional group right here. Notice that you have this group right here, carbon number 6. It's above the ring. So it's almost like if the ring is almost flat, and you have carbon number 6 above the ring. ماشي? Can you see that? Now, أعيد. Right. So you can have carbon number 5 of glucose attacking, forming a covalent bond with the functional group, which is carbon number 1, forming a ring structure. This ring structure, the name of the ring, is known as a pyron ring. Because it's a sugar molecule, we add OSE to the end of it, so we say that's a pyranose. منيح? Okay. So if I say a pyranose, على طول بتعرف this is a sugar molecule that has a pyron ring, and this is the pyron ring, when you have oxygen as one of the components of the ring. Now, and it's drawn in a way where it's given this three-dimensional feel for it. So it's not like you're looking at it two-dimensionally, rather it's a bit tilted so that you can see it in 3 dimensions. So that you can see that carbon number 6 is actually above the ring. You can see that this OH, this hydroxyl group, is below the ring. ماشي? You can also have glucose molecule. You can have carbon number 4, the hydroxyl group of carbon number 4 attacking the functional group, forming a ring structure known as a furan. So this is a furan, a 5-membered ring, where one of the components of that ring is an oxygen. So that's a furan. Since we're talking about a sugar molecule, we say it is a furanose. Yes? Bordeaux is given this three-dimensional feel for it, and you can see that these two groups are above the ring, and this group is below the ring. معاي؟ نمتوا؟ لا لسه احنا ما دخلنا بالمحاضرة اصلا. ماشي؟ طلع ring structure. You can see that this group is above the ring, this group right here is below the ring, this group is below the ring as well. هذه above the ring. Same thing for this right here as well. Which one do you think is more stable? The pyranose structure or the furanose structure? Pyranose. So most of the sugar molecule, the monosaccharide, glucose or mannose or galactose, they exist in the ring structure. Not only that, they also mainly exist in the pyranose form rather than the furanose. You know, pyranose is more stable. Notice something. Once you have this reaction happening, the idea, by the way, is that this group right here, this carbon right here, is actually a hemiacetal. This group is a hemiacetal, so that you can have a carbon that is linked to OR, OH, C, and H. So that's a hemiacetal. Now notice something. Now once you have the formation of this ring structure, all of these groups are fixed. They do not move, except for one, which is the one that is linked to this carbon right here. So this hydroxyl group can either be below the ring, or you can have rotation of this group so that it flips and goes above the ring. So you can have two forms of that ring. You can either have the OH above the ring or below the ring. Now, ايش هو هذه dynamic. All of these are fixed. خلاص. They do not move. This OH would still be, would always be below the ring. This OH would always be above the ring, below, and so on. Except for this hydroxyl group, it can either be below the ring or above the ring. These two forms of a sugar molecule in the ring structure are known as anomers. So if I say isomers, بنعرف ايش يعني isomers. إذا بنحكي diastereomers, عارفين ايش يعني diastereomers. If I say anomers, على طول it comes to your mind, this is a sugar that exists in the ring form, one, and the OH is either below the ring or above the ring. Now,
دكتور هلا الالفا جلوكوز هو نفسه can be converted.
هلا استنى شوي. Yes. هلا these two anomers are given a designation of alpha or beta. If the hydroxyl group is below the ring, it's known as alpha sugar. If it is above the ring, then we say it is a beta sugar. To differentiate between the two, alpha is drawn like this, right? That's alpha. It looks like fish, and fish swim down below, right? In the sea. Beta is bait, and bait, you build it above, on the ground, right? On top. You don't except Amman. You don't build a house underground. You build it above, right? So it's beta to differentiate between alpha and beta. So right here you have alpha, the fish is right below, and here you have beta, where you have the building up there. Same thing with fructose. Again, we're looking at the functional group. We're looking at this carbon right here, and this carbon, we call it the anomeric carbon. This carbon, again, we call it the anomeric carbon. So if you look at the anomeric carbon in fructose, fructose is a ketose. So you have formation of a ring structure, and you have it with carbon number 2, which is the functional group, the ketone group. And you look at this group right here for carbon number 1. It's below. If it is below the ring, then it is We look at the OH group. If it is above the ring, it's beta. If it is below the ring, then it is alpha. Same idea. So that's how we can differentiate between alpha and beta. Okay?
سؤالي كان إنه مثلا ألفا جلوكوز ممكن بعدين يصير كيف بالحالة تحول لبيتا جلوكوز.
Oh yes. This process is known as muta-rotation. Muta يعني mutation, rotation. So yes, alpha can change to beta. So it's dynamic. It always happens. طيب.
نحكي الجلايكوز لما نحكي الجلايكوجين مثلا ألفا جلوكوز مونومر.
نحكي بعدين بقى. يعني one step at a time. الله معك. Please check the listing of your seat number. طيب. لما نبعث لكم إياه.